We'll begin with the structure of antibody, and I'm going to talk about the properties of antibodies, some of the methods that we use, different types of antibodies, and then also keep in mind when we talk of antibodies, we basically are talking of a therapeutic agent. We're talking about something that is going to be the ultimate therapeutic agent for treating quite a few diseases. They have started being marketed up, so it's good for you to basically have a good grasp of this information. And again, it's a little bit long chapter. I'll see if I can finish with one, otherwise I'll have to do in 2 lectures. And again, I did post you the link that I had for the Wiley book. And the more I read the book, the more I like it. It's really readable. It's good. So if you remember, it's tough to teach a boring subject like immunology as compared to an interesting subject like biochemistry, which you guys like, I heard. So I'm going to try to make it fun. And one of the things that I would want you to do is that we're all different. We may have different styles of learning, but to make a mnemonic, if you come up with an idea, hey, this is how I remembered it, instead of keeping it to yourself, share it with the rest of the community. The only condition is that it should not be vulgar or too much obscene, because quite a few mnemonics are like that. People tend to remember bad things more than good things, probably. Anyway, so one of the mnemonic I told you, and I'm going to have all this just looking at watch. When I close the lecture and finish the lecture, everybody starts getting up. They don't even want to look at the watch at that time. When it anyway, that's fine. So you didn't miss much. Anyway, so if you do have a mnemonic, please share it with me, and we'll try to come up with something interesting, something to make it a fun activity. Because research does show that people tend to remember things when it is in context with something that you want to learn about, and something that you want to retain the information, which is extensive, which may be boring at times, and you may or may not like it. So if you remember, one of the things I said that just for mnemonic purposes, if antigens are boys, then antibodies have to be girls, right? So I'm going to use all these, similitudes when I teach you different concept, and these are all mine. So if you want to change it, learn a different way, by all means. And if you find something better, share it with me, and I will pass it on to the rest of the people. And those of you who are interested, I have tons of mnemonics from my previous 8-9 years ever since I've been here in this school. People have come up with all sorts of study guides, exam guides, tables, mnemonics, so on and so forth. I don't want to overload you, but they are available. So let's assume, so today is a girl's day, in that sense. How many of you have seen this movie, Incredible? Well, you're too young for that, right? Okay. Okay. The point I'm trying to make in this one, and I really like it, that the writer, which is Brad Bird, and he's also a director, and he's saying that just want to plan, something you plan in your mind. And in this case, the dad expected to be strong, so he made him strong. Moms are pulled towards in a million different directions, very interesting. So he made her stretch like taffy. Then there are teenagers, boys and girls. But each one of them has a particular kind of, acceptable behavior or point for you to remember. So you can kind of move that to a boring learning of conceptual things. That's what I want you to do. And that was an exercise we gave to the people last year. And then again, 10-year-old boys, hyperactive, babies are unrealized potential, and so on and so forth. So what I told them to do is that they can go for all character. And you can also characterize the different cells, immune system, a T cell, a B cell, a dendritic cell. So it's going to stick in your mind. I remember now when we were taught immunology, of course, as I said, there's too much content. I used to say it's a French, and I don't know French. So you need to associate. It's called association memory. So it's not going to be what you call vomit out thing that you remember it for some time, vomit it out in the exam, and you're done away with that. That's not the purpose. So try all these characters. If you can find something better, let me know. Okay? So I'm going to use that today as antigen as in a boy and antibody as in girl. So you can see from here the structure of an antibody, and I think what I'm trying to say is that a little bit of anatomy. If I was to tell you that there are heavy chains and light chains, I'm going to say there are 2 heavy chains and 2 light chains. I'm going to put it in your mind like 2 heavy chains as legs and 2 light chains as hands. And then keep in mind what does the hand do and what do the legs do. And then I also want to keep in mind, especially in this person, the ability to move, limited ability to move. So in the shoulder joint, you have like 360-degree rotation. You can kind of rotate it 360 degree. Well, if you're an athlete, you can do it. Hip joint, 360, but I don't think each one of us can do that. I'm pretty sure there are people, right? And then ability to hold on to things with the hands, the ability to stay on the floor with the legs and foot that you have. Okay, so start kind of synthesizing the information in your mind, thinking, I'm going to learn structure of an antibody. So let's see a nice fit girl. And then you look at the contrast she has. I'm pretty sure men don't really bother much about the contrast. But she has red. So you see the combination. So that's the important thing when you have to learn antibody. That's why I didn't say antibodies are male, because I know the male behavior. They don't really care for all these details. I think most of them. But the females are very particular about that. So that particular behavior is one of the top quality of an antibody being a drug, specificity. Specificity, very important. I'm going to discuss that in detail. It will be unusual or crazy for her to wear 2 different type of shoes, or it's like more of a color, more of a synchronous activity, more of a fashion, so on and so forth. Okay, so let's move on. So antibodies, if you remember 2 important things, humoral immunity. I said cells are important, so keep that in mind. A cell, right? And then in the blood there are cells and there are some proteins. So antibodies are not cells. Okay, I don't think there's any argument on that. Isn't it? Is there an argument? Okay, so they are proteins, that's for sure. And the important thing is that they're soluble proteins. Very good. They're soluble proteins. We want soluble proteins. And then they are the major line of defense. And most of us, when we give vaccination, as I said earlier, we are giving you antibodies. We also give you antigens, but that's an active immunization. When I give you prepared antibodies, we give you passive immunization, prepared, cooked food. Now, they contribute to immunity and protection from foreign material. As I said, yes, they do. Then they are from a class of proteins which are globulins. So you have this albumins, right? You have globulins and so on and so forth. If you learn them electrophoresis, you'll see they kind of settle down according to their weight, right? So lighter will travel more as compared to heavier, right? So they are globulin per se. And then since they are part of immunity, we used to call them immunoglobulin. Now, when I say they are soluble proteins, also keep in mind, and again, I want to stick in your mind 2 important concepts, that these proteins either are free afloat or they stick. Okay, so I want you to keep in mind, like that girl, that she is going to stand on the ground. So she has foot sitting on the ground. So that ground being a cell membrane, so she's going to stand over there, right? but it can be free floating as well. So two types of antibodies. One are floating, right? And one are membrane bound or membrane unbound, whichever way you want to put it, right? It's a very important concept. Okay, and if you remember yesterday, I told you that B and T cells are two important cells, and both of them have receptors. We call TCR, T cell receptor, and we call BCR, BCR receptor. Then I also told you a very special thing for the BCR is that BCR are antibodies. Very good. Very good. BCR are antibodies. So we're going to use alternate. Either we'll put BCR, we'll put antibody. I haven't told you the structure of TCR as well, but it's going to take me a long time to explain that to you. But keep in mind, when I talk of antibody, I'm also teaching you BCR, which is a B cell receptor because they are antibodies. Good, very good. Now, also keep in mind that these are produced. Now, another concept, again, with the incredible team that I have for you today, mom and dad. So if I was to make that mom and dad is strong and mom, whatever capability she has. So I said, okay, let me make T cell as dad and B cell as mom. Okay, so keep that in mind. It's going to help you. So T cell strong, powerful, there's a big mighty person, main bread earner, so on and so forth, and the B cell. Okay, and also if I was to ask you that, the mom and dad, the kid's going to come. So who's going to bear the kids? Mom. Okay, so if I was to tell you that antibodies are being produced and born by which type of cells? B cells. Okay, so B cells, they will produce these antibodies. If it's a family where, and I'm going to talk about antibodies, and I've got quite a few. I usually, you'll see me in rest of the micro and immunology, I usually come up with some kind of impact slide. I want to have you visual thing in your mind, and you can kind of draw because notes are incomplete. So you keep on writing things and whatever you want to do. So a B cell, and then again, don't take them literally. Whatever mnemonics I tell them, don't take them literally, just for explanation purpose. Don't say, in the University of Chicago rotation, my professor told me T cell is a dad, and you're not supposed to do that, right? So the B cell produces, B cell mom, she's going to produce antibodies. So antibodies in this family will be daughters, correct? So that's another important thing for you to learn, right? And if I may use this term, if I was to call this B cell which is about to produce antibodies, I'll say pregnant B cell. And there's a term for that. We call that plasma cell. So the only thing I'm trying to put in your mind is that there are two types of B cell. One is a B cell is about to bear kids, antibodies. Right? So this is called plasma cell, as compared to the other B cell, which is just a plain B cell. Okay? Now, that's what I say. Plasma cell, terminally differentiated B cells in bone marrow. That's what it's saying, it's about to produce antibodies. I told you two important features, especially in terms of me picking up as a girl, specificity. Very, very specific. They're specific for everything. They eat, rest, whatever. You know that. So that's a quality of an antibody. And then the important thing is that they have a very intense biological activity. Their participation, like moms, you cannot run a house without a mom. So everything has to go through there. So she is all in all. So she has all the functions of antibodies that we have. Okay, so keep that in mind. And then again, if I say functions of antibody, I'm going to use another mnemonic, which basically may not be here, but it's called PAIN, P-A-I-N, PAIN. P for precipitation, and A for agglutination, and I for immobilization, and N for neutralization. And this is the basis of all the lab tests we have today in the market. All antibody based. Okay, mnemonic PAIN, so you don't remember. And you can see over here that they are precipitation, agglutination, immobilization, and neutralization. And that's very important biological activity done by these special molecules that we give you as injections. We want to give you treatment. Now, as I said earlier, they are present in serum. And also keep in mind that those of you who are downloading lectures from Moodle, I mean, since I'm reading as well, so I'm a little bit tweaking things here and there. And hopefully, I'm still working on these current lectures to be posted onto whatever web hosting thing we can come up with. But they're with me. If you're really desperate, you can always come. They take a lot of space. We have some issues. We have some colleagues of you working on that. But keep in mind that there's a little bit of difference, and I'm adding things up. But again, if you are reading book, you should be okay. So if you run them, so it's an antibodies containing portion of blood. That's what we say serum. Is one of the definitions of serum. Is the liquid portion of the clotted blood. Liquid portion of the clotted blood. So this is something should be there in your mind. If you run them on electrophoresis, and again, these just immunoglobulins were run on an electrophoresis, and I'm going to give you, there's a example coming up with this mom and dad and family. And so this is a family, I usually call them a GAMD family, GAMDE. We'll talk about that later. So mom and dad. So they have 5 daughters, okay? And each of them are unique, so I'm going to share with you their private medical history. So you tend to kind of remember them. So you need to remember all these daughters, which are antibodies, separately. They come from the same mom and dad, but they are unique. Okay, so if I was to ask these 5 girls to run, so obviously the lighter one will not have your, okay, so keep that in mind, keep your mind open. It will be easier for you to remember. So slowest of them all, that runs on a journal, now I'm talking about all the plasma proteins. In this case, I'm talking about all plasma proteins. You take the portion of the blood, which is the liquid portion of the clotted blood, make it run on electrophoresis. It's going to divide into alpha, beta, and gamma proteins, which are plasma proteins. And gamma are the slowest of them all because they are heavier. Okay. Now, also keep in mind that these antibodies, I gave you like a structure for that, are to be produced, they are in the blood. They are to be produced in the body, and things can go wrong. I just picked up one of the pathophysiology over here. In this case is that if you have a disease which is called multiple myeloma, where there are more proteins, and you may have some of the proteins being secreted in the urine. So these are components of these antibodies. They either appear in excess, so they are norm level. They're normal levels of each and everything. So each one of us has normal protein levels. So they can go up and down. If they do so, then we are in trouble. It can be cancerous, or it may have some other difficulties that we have. But just remember 2 important concepts, which is multiple myeloma proteins and Benz-Jones protein, which appear in urine. And I'm just going to present that because that is there in the book, just to give an idea. We kind of tend to teach you normal, abnormal at the same time, just to kind of give you an importance for that. Now, if you were to run all these proteins, as you can see from here, on electrophoresis gel, all the serum proteins from a normal individual, and go from cathode to anode, and you can see from here level, and you will see these albumin and globulin and other. But what you will see over here, like blue, is a normal, right? And then there are some of the proteins that are excess. So these are like myeloma proteins. Well, it's okay if you have a little bit of excess, but we have a problem over here when we have a gamma portion which is way up. And why do we care for the gamma proteins? Because they are antibodies. They are the gamma globulins. The others are globulins. They are albumin, and other proteins which are there. But these are gamma globulins, and that's alpha, beta, and gamma. because then we have a cancer. We have a problem here, overproduction of cells, uncontrolled production of cells, and that's what you see on electrophoretic mobility, that you see that as a diagnostic test. Okay, now let's talk of things in detail. Also keep in mind, first study was done in 1959, and then I've got all these numbers. And the important thing, as I said earlier, most of the immunology, I've got this text just to kind of tell you where is it written in the book, but most of it is a visual thing. So you really have to have it in your mind so you can relate and draw and kind of help yourself. So let me go to the next one, because that will be the explanation of this one. Okay, girl stuff. Don't ask me. I don't know anything other than what I'm kind of trying to make up over here. So if the legs are heavy chains, right, and the arms are light chains, so 2 light chains and 2 heavy chains for every antibody, right? But these are 2 legs, but remember, they have a different footwear, right? I'm not going to tell you, one of our professors was telling me that she had at least not less than 200 pairs of shoes. I thought, oh my goodness. So antibodies are like that. 200 is too little a number for them. Okay, but in this case, what I'm trying to do is that there are 5 different types of shoes that this basics antibody structure can wear. So they pretty much are like 5 daughters. They are all daughters, right? But they are different. So we give them a name based upon the shoes they wear. That's funny, but that's the way it is. I'm trying to explain that to you. Why I'm telling you? Because the difference is in the terminal part of heavy chain. That's what I'm trying to tell you. If these legs are heavy chains, right? So it happens to be the terminal part. That's where you put on different shoes. Does that make sense? So in this case, M, or let me go for G. Let's go for G, because G is a gamma. So there's an antibody named gamma. We call it IG. IG for immunoglobulin, IgG. And why we call it G? Because the terminal part of the heavy chain has gamma subunit, right? Then we have A, alpha subunit, right? Then we have M, micro subunit. Then we have D, delta subunit. And then we have epsilon. So GAMDE, that's what's a GAMD. G, IgG, IgM, IgA, IgD, and IgE. They all are antibodies. They all have the same structure, little bit of difference, and that will make enormous difference when we go and go for some other things. So far, so good? Okay. Now, let's go a little bit biochemistry. You can see proteolytic digestion. So if we have this structure, 2 arms, 2 legs, and now you can see from here, the only thing that you have to put in your mind, and I cannot come up with any other, in reality, if I was to make that similitude between this girl and heavy light chain, you have to keep in mind that that particular girl, or myself, if I'm an antibody, my arms and my legs, they are like heavy chains. They go all the way from the top to the bottom. And then there are additional arms on the sides. Additional arms on the side. They basically are light chains. Okay, so now, if you look over here, these ones in the center are heavy chains, right? And I'm still going to use the similitude of arms, and I'll explain that to you why. But these are like extra things on the side. They're attached to the upper portion of the heavy chains, which we call light chains. Okay, so in theory, as if you have 2 arms on the right and 2 arms on the left with 2 legs. So this is like a biochemistry aspect. So these are heavy chains, and these are the light chains, okay? and they are joined by disulfide bonds. That's another important thing. They are joined by disulfide bonds. And what we did was that we had two enzymes, papain and pepsin. So what happened is, if I was to use papain, it's going to cut here. It's going to cut here. So you have 4 arms on one side and 2 legs on the other side. If you were to use pepsin, it's going to cut over here, where the upper 2 arms will still be united with the disulfide bonds, and then the legs basically will be loose. So keep that in mind, and I'm going to introduce 2 important terms right now. The whole idea for this experiment is that we are fragmenting this protein structure by using enzymes. Okay? So if I to use an enzyme to fragment 2, and I'm going to use 2 fragments, FC, right, which is over here, right, and then the other thing I'm going to use is a FAB. So C stands for constant fragment, and FAB, AB stands for antibody binding. So this means if this antibody was me, I want to hold anything, I will always hold things with my hands, because my hands are FABs. Does that make sense? Whereas the constant portion I have are my legs, which are constant. So we will have, so if I was to ask you, how many constant portion in an antibody, so you'll say 1. Or how many constant portion in a FABs in antibody? 2. Right? But at the same time, if you look at valency, you understand what valency is? Okay? In valency, so you have 2 constant portions, and you have 4 Fabs in terms of valency. That makes sense? Basic concept. So subfragments, constant fragment. Now if I was to ask you, which one of these segments is going to attach to a membrane? FC. Because I just told you, like legs, you have to stand on the ground with your leg. FC. Right? But also keep in mind that you can hold things with your, which is called antibody binding site, FAB. You have to write it on a piece of paper and give it to me. I'll take question in that. Okay? Sorry. All right. Now, again, this is a schema of that. You can see from here in terms of biological activity. I gave you an example over here. This is a constant portion. Right? So I just wanted to inculcate in your mind the constant portion will basically be so let me put it this way. So if I was to ask you Fab, so Fab will have a constant portion, a heavy chain, and the light chain. Whereas FC will only have heavy chain, no light chain. Very good. Simple. Okay? So that's an idea from here. So you can see these disulfide bonds, but these are basically, okay. Now, remember I said FAB, antibody binding site, but also keep in mind, if I was to talk of antibody, this is the site where antibody binds to something. So to something is an antigen. So some books will say antigen binding site. Does that make sense? So it has a dual meaning. So I would rather go for antigen binding as compared to an antibody binding, because antibody is a part of antibody. It's a part of antibody. Over here, you see antigen binding. And the idea is that if antibody has to hold onto an antigen, it's going to hold with the hands. So these hands basically have 1 light chain and 1 heavy chain. Does that make sense? It will hold antigen binding. And then again, as I said, example for the upper arm as hinges, so they are hinges. And then there are carbohydrate moieties. We talked about FC portion, we have for FAB. So these are the 2 basic concepts that you have. The other important thing we call mostly in terms of constant. When I say constant, so obviously there should be a variable portion as well. If I say constant, it's a variable portion as well. So in this case, this part over here is variable. So if you see like this blue, which is basically a constant portion, okay, and then the distal part of Fab is variable portion. Okay, and I think I'm going to give you another mnemonic, whether it's coming up or not, I don't know. It's just like this girl, she puts on different shoes, but she also puts different gloves. Right? So our hands are the same, but she puts different gloves. So these gloves are variables because she can color, match, whatever she wants to do, right? So this means the antibodies may have a variable portion for different antibodies to hold on to things. So if I'm going to hold on something, I'm going to put on gloves. So that gloves basically are here, which are variable because I can take them off and put them in. But I cannot play with a constant portion because this is constant. Okay, I'm going to explain that to you again when it comes to the next slide. So I already told you about the constant portion over here. She's holding up with these dumbbells, right? And then there's a flexibility over here in terms of movement. Okay. Now, the other important thing that if it is an antibody, right, it has a tail piece, we say lead, and that's where the tail piece is going to go and attach to the cytoplasmic membrane. We already know, right? Now, the other important thing is that when I say this antibody has two hands, so this means that if I was to pick this up, I have a hinge joint, so I'm going to use both hands to pick like this, right? But if I was to pick this table, so I can extend my arms over here to a certain limit, I can pick this, but I cannot do that. So there is a physical restriction on this hinge joint for me to pick up an antigen. And you see over here, if two antigens are located close, the antibody is going to go and pick them up like this. But it can move to a certain degree. Okay? So that's another, we call flexibility of antibody molecules in terms of they kind of latching onto two different antigens. So that's another important thing because I'm just building up the whole sequence for you to understand. Now, when I say variable, so variable like gloves, if they're all proteins, right? So if you were to run the variability of the amino acids for the variable portion of these proteins on the end, distal proteins, you will see that they will fall into different amino acid positions. So the amino acid positions or amino acid positions basically are the one because they're proteins. It's just like, oh, each one of you have the, different cell number, but the numbers are staggered here and there. So you end up with the same amino acid sequence is changed. So that is where, when we look at that in terms of the immunoglobulin molecule, you will see the mostly variability is in CDR1, CDR2, and CDR3. Just have to memorize it for now because tons of amino acid. I'm going to go in detail later on. So the distal part of it is that amino acid sequences are different. That's why they are different because they are proteins. Okay. Now let's see. For example, there's an antigen. So the antigen comes to your body. Like, for example, you've been vaccinated with hepatitis B. They gave you like just give an example. Or let's take an example of MMR. So you already made an antibody in your body against these three viruses. So the moment these viruses are going to enter into your body, like let's say they have an antigen, and you already have antibodies. So antigens, as I said earlier, are a big chunk of molecules, compounds. So they have different sets of these epitope structures. So depending upon the size, because size matters now, because they are immunoglobulins, right? So they are immunoglobulin. They have definitive size. They have molecular weights. I'm going to talk that in detail. I want you to know that. But I'm just coming over here. So there are three different types of, for example, heavy chains or light chains. They can come across these antigens, you can see from here, and see if they can complementary bind them. Because it's like antibodies, with head and tail running in the fluid, and antigen comes, so they're going to go and look around and try to bind them out, right? That's what you saw. The biological quality is just like, bad example. So just wanna, if a person is making problem, so one of the things the policeman will just kind of put him down, so that just control it. So that's what the antibodies do. If antigen is causing problem, they all jump onto them and immobilize. So no damage is done right away. That's what they want to do. But before they would do that, they're going to look for all these places. But you can see from here, in terms of sequence of amino acid, like for example, this one has 54, 55, 56. They are all different, right? So firstly, they're going to go, that's why we say the antibodies are very specific. This means that if you give MMR, which are T3 viruses, they produce antibodies which have these three, so they will put on those gloves, and those gloves are only for picking up those particular viruses, but they have to match. If they don't match, the antibodies are useless. That's what specificity is. So you can see right now, they are looking for that. Okay, that's why the specificity is out. Now, I tried to, it's there in the book. I did get a PowerPoint presentation from them, but unfortunately they didn't give me the tables. But the only thing is that I would want, I've summarized it in another picture, but I would want you to read in the book, and I got it over here. So what it's telling you is the features of, the factors and features of IgG, A, M, D, E, all these features, and what are the differences. And that's what I want you to know, and that's what I'm going to spend next half an hour or 20 minutes, whatever I have. Okay? And they are all important. We want you to know, objective of today's lecture is G, A, M, D, E separately. We want you to know this family in detail, and I'm going to try to do that. Okay, now, one of the concepts is like specificity concept, basically is same, we've been talking over on lock and key. So if there is an antibody and there is an antigen, so it's going to look at it, right? There are two ways that they can lock in. One of the ways that this antigen can lock in, it's going to see over there, go into the groove. So what may happen is that sometimes the key goes into the groove fits in, but it's not going to open. Some of the keys will not even go and penetrate. But in this case, what's happening is if the two antigen 1 and antigen 2, and these are antibodies, they want to come and look at them and bind them. So in this case, both of them basically show the binding depending upon the external epitope. It's like, that best fit that they have over here in terms of the structure homology. So that's again something you have to, that's where things go wrong. Remember you said cross-reactivity. So we made this antibody for this antigen, and we had a good faith in that. But this also bound to this one, which wasn't supposed to do that. So you may have problem here. There's a cross-reactivity based upon the antigen structure. Make sense? Okay. The other thing that you can see in this case is, now interestingly, you have an antibody, right? And it is supposed to bind with an antigen, right? But I'm going to give you another concept over here, and now things are going to get a little bit complicated. If antigens are all compounds that evoke an immune response, and the worst form of compounds are proteins, having said that, antibodies are also compounds and they are also proteins. They can act as antigen themselves. Okay, give you an example. So normally it is an antigen-antibody combination that causes immune response, right? Now let's say you have an antibody and this should bound with this antigen, perfect fit, but all this binding and everything is based upon sequence of amino acids. Now in this case, what happens is there is an other antibody, which is an antibody against A, that snugly fits over here. So once you have this lock and key, you will open the door for immune reaction regardless of whether it's an antibody. So this basically now acts as an antigen-antibody complex. The other example over here is that you still have an antigen that binds to an antibody, but there's some exposed surface of this antibody that your body recognizes as foreign. So it's going to make an antibody against an antibody. Because that antibody is a protein, and that protein has an amino acid sequence, and that amino acid sequence is foreign, so body is going to mount an immune. That's why you have an antibody against an antibody. Because these are functional things. So antigen, antibody is a functional thing. That makes sense? So this means is that this antibody has a sequence of amino acid. I gave you an example. A sequence of amino acids, right? So there's a T cell over here. T cell comes over here and say, hey, these don't belong to me. Let's make antibodies against them. So you're going to make antibodies against them. So you can see you have an antibody B. In the blood group, for example, you have an antigen A blood group. But you have an antibody against B. But if you have an antibody against A, and you have an antigen of A, you have a problem. So these terms that I'm going to use, antigen, antibody, that's why the better term is immunogen. Because when I say immunogen, it doesn't mean antigen. It can be an antibody itself. All immunogens are antigens, but all antigens are not immunogens. So that's a concept you have to have. Okay, now let's go to the different types. And I said the 5 isotypes. I'm going to go all that, and they are, those of you who know genetics, I'm going to give you a little bit of genetics over here, allotype, idiotype, specificity, and so on and so forth. And it will be good experience for you to go and read it up as well, because I don't want to waste my time to teach you the basic genetics. Now, these are different types of immunoglobulin variations that we have. So when we say isotypes, so what I'm trying to say is 5 sisters. That's what I was telling you. 5 daughters, 5 sisters. G, A, M, D, E. Okay? And the difference that they have, why this is G, because the constant region is G, because she is wearing that shoes that basically give her whatever she is, right? So that's attached to the constant portion as compared to another sister, they look alike, but the constant portion over here on the heavy chain is an A. And remember that it's nothing to do with the variable position. It's nothing to do with variable part. That's why it's called isotype. So if I was to ask you isotypes, I want to hear G, A, M, D, E. The reason is because they have a different subunit over here in terms of the sequence of amino acid. So if I was to say allotype. So what I'm trying to say now is that within one family IgG, so they both are IgG, the blue one, there may be subtle differences over here in the sequence of amino acids, right, which will give them a different name. So we may say this IgG1, IgG2, IgG3, because they're subtypes of the parent type. And if you talk about idiotypic differences, so what we are trying to do is that we are wearing different gloves. It's called idiotypic because now the difference is in variable portions of both heavy and light chain. Okay, let me give you another example. Same slide. This is the same. So that's what I'm trying to tell you in this one, that when I say isotype, I'm talking about GAMDE. Differences result from the structural variation in the heavy chain. Structural variation in the heavy chain. As compared to allotype, where there are genetic differences, where you have different genetic allele causing one or two amino acids to change of a length of the amino acid, and that will have subtype of the parent type. Okay, as compared to idiotype, where differences between the constant regions due to expression of different heavy and light chains. So you can see for me, I'm going to go and explain that to you in a while again. But this is something idiotypes that we normally make, in the lab, and we're going to use for the drugs and so on and so forth. Now, this idiotypic difference results from structural. So this one is same gene at a given locus, distinction within a species. So if you say, like within a species, all males and females, so they have similarities within the same species. As compared to idiotype, where you have different allele of the same constant region gene. A little bit of genetics, but I'm going to come back to it again. But I'm pretty sure you must have realized the easiest part for you to know is isotypic differences and allotypic differences, because that's pretty much easy. This is a little bit challenging for those of you who have not taken genetics, but I'm going to come back to it later again. The concept I'm trying to get is the gloves. Just like, you see that structurally, they all look the same person, but different type of gloves. That's what the whole idea is, right? So the binding portion, why I say glove, gloves is something that's going to touch. If I put on the gloves, I'm going to touch this. I want to protect myself and protect this. So that gloves is something additional. So that's what the specificity comes in terms of, I'm going to see each and every patient by putting on different gloves. Pretty much it. I don't want to use the same gloves for handling the same person, same food, so on and so forth. So this is on top of my arms, which are pretty much the same. Hands are the same. So that's why we got variable. You can't throw off your fingers away and, hey, I'm going to try to new. No, but you can throw off your gloves and put on the new gloves. Okay. Now, again, you can see that different types of. So what I would do is I will leave these charts for you to read and give you a mnemonic in the next couple of minutes. Some mnemonic, so you can go back and try to kind of, strengthen the concept over here. So let me do that. And this basically, these are the properties of, I just told you, the pain, and the different types of antibodies. And these are the basis of the concept that it may look difficult to you to remember right now because too much cramming, right? And they are important as well, because I would want you to know, for example, what antibody crosses epithelium. I want you to know which antibody crosses placenta. I want to know which are antibodies which are present in intra or extracellular place. I want you to give me the serum levels of different antibodies, where and how. So these are all functional elements of the 5 groups that you need to cram and you need to remember. So they're all tables full of that. I don't want to go and read it out for you. But what I'm going to do is I'm going to give you for today for over the weekend to work on is a similitude, a concept over here. And that concept I said is called GAMD sisters. And you can see gamma, alpha, micro, delta, and epsilon. And this is a chart that I made. So if you kind of try to remember this chart, what I'm telling is that in the family history, there are 5 sisters, and there are their ages, date of birth, and then different things which are there in terms of their personal medical history. And I've just picked up all these things for a good purpose. Okay? For a good purpose. And then in the family record, if you go over here, the B and T cells are mom and dad, and then they have aunts, which are basophil, eosinophil. Mast cells is one of their aunts. She's a pharmacist. That's interesting, right? That's the only interesting thing. And then again, macrophages are called big glutens, neutrophils are called traps. So these are similitudes, your books are again. So in this book, they say dendritic cell is an FBI agent because they keep all the data files for you, unfortunately. And then they have brothers as well. The brothers are CD4 cells. And these are some of the family traits their father told them. Don't talk to strangers. Because they're antibodies, they're girls, right? They don't react with anyone. So there are restrictions there, okay? So let me give you the, you can draw it up. I think it's gonna come next slide. There you go. Okay. Interesting, right? No? You're not interested? Okay. All right. Now, 5 sisters. I mean, you will wonder why, how did I find all of them at standing for me? Okay. Don't have to take photograph, I'll give it to you. You can take my photograph if you want. Okay. Okay. So, G, A, M, D, E. 5 sisters, okay? Now, G over here, so they all decided to go to gym and they want to show up, the kind of, fitness. So it's all a fitness exercise because New Year, whatever you want to say. Huh? New Year what? There you go. So the first one over here, as you can tell, she is a professional. And she is very versatile. And she has a driving license. That's funny, but I'm going to tell you why did I tell you that she has a driving license. In my story, they don't have a driving license. So bad. Now, the second one over here, the sister is, I don't know, you agree with me, a little bit chubby, but she's a swimmer. Okay? The third one, you can read it for yourself. Okay? She's a big hugger. I'll explain that why I do that. And then we have mom's baby. She's the youngest one. So she's a mom baby. She's very close. They kind of hang around together. Right? So mom baby was just like a boy of the whole group. And she has an attitude. She wants to stay away with the rest of the family. A little bit, I can tell by her face. Allergic, and she has good friends with one of the aunts, which is mast cell, and that happens to be a pharmacist. So she said she would rather spend more time with taking drugs for getting whatever body they want to get, rather than doing exercise. This is her philosophy. She's unique. So they just joined this health club, right? So she kind of spends on average 23 hours per month, and you can tell what's happening here. She spends 5.5. She spends 5, and you can see from here. This one, again, I told you that she wants to rather take drugs to develop body instead of doing an exercise. So they're a rich family, richer than the instructors in pharmacy school or maybe pharmacists as well. I remember one of my students came to me, 2 students came to me and said, they offered her 120 and they offered me 110, and I think that's not justified. So I said to myself, oh my goodness, my students get more pay than myself. So I would have joined a pharmacy school and be a pharmacist than to become a professor. But they're a rich family. And this is their income breakdown: 150K, 160K, 900K. So she has a good reason to be what she is. You have to spend money. If you earn, you need to spend it. 180, good enough. The other characteristic for this family is that both of them are very fond of milk. Did you notice that there were only 2 there wearing the shoes? Right? A good reason, because they love proteins. Keep that in mind. They love proteins, so they're into proteins. Okay? Now, all those tables that are there, I'm just going to get a couple of minutes extra. Is it okay? All right. So what I'm trying to say is that if you go back, keeping this in your mind, that each and everything tells you something. Each and everything tells you something, right? For example, this one is the half-life of these antibodies in the blood. So if you try to remember in any other way, I don't know, you can come up with a better mnemonic, but I guess it's not going to be easy for you, right? This is IgG, so that's a professional and very versatile, the most important antibody that we have in our body. And it exists, and I said both of them are homebound, so they are all intravascular. Okay, except that she has a license, so she can drive out because she has extravascular presence as well. So that's the only antibody that exists in intravascular compartment and extravascular compartment. Okay, and these are their molecular weights. Okay, now this one over here, she's big, right? So she's like, so she wants to make sure, so she wants to attach to others. It's more of a attaching. So that antibody has this ability to attach, and I'll explain that to you why that is. But for now, I just use it as a hugger. And then again, she has a sweet tooth. So this means she wants to bind to complement. This is the only complement binding protein. And complement is sweet. This is the only protein that is present in serum. That's why I call she's a swimmer, which is serum is liquid. These are the only two proteins that need additional protein chains. That's why they put on two shoes. No other antibody would need an extra protein chain except A and M. These are the two proteins. They like milk because they are secreted in the milk. If I was to ask you which are the two proteins secreted in the milk, guess what? Only G and A. When I said that she's an attitude, so she is allergic, so this means IgE is the major antibody that is for allergy. If I ask you, the other name for this antibody is allergen. Because that, if you say, if you are allergic, I'm going to run your IgE antibody levels. Right? The other thing is that these IgE, so let me go over here. She is mom's baby, so she want to be in the lap of the mom. And guess what the mom is? B cell. So what I'm trying to tell you is the IgD attaches to B cell. That's the function of IgD. This is the only antibody that attaches. The only other antibody that can attach to any other thing is M, because she's a big hugger for everything. Does that make sense? Now, this one, I said that she has an aunt, she's a pharmacist, she wants to go, and what's the name of that aunt? In the previous, I said the Gamma family, mast cell. So this is the only antibody that attaches to the mast cell, and that's what causes allergies. E, that's what I said, allergic pharmacist, right? That makes sense? So each and every, it's just like how you create a pneumonia and kind of put them together.